Have you ever wondered how you can suddenly find the energy to sprint for a bus? Or, how your brain keeps firing on all cylinders, even between meals? Well, in this explainer, we are unlocking the secrets of your body's personal power grid. We're going to be talking about glycogen, which is the crucial energy reserve that makes all of that possible. So let's get into it.
So here's our roadmap for today. First, we'll get a handle on what this energy bank is. Then, we'll look at how you make withdrawals for quick energy, and of course, how you make deposits after a meal. We'll also explore the brilliant management system that keeps it all in balance. And finally, we'll see what happens when that system breaks down. All right, let's jump right in. We're starting with our energy bank. We're going to build up our understanding of what glycogen actually is and why it's so incredibly fundamental to how our bodies work. I mean, really think about that for a second. When you need that sudden, powerful burst of energy, where does it actually come from? It feels almost instant, doesn't it? That's not coming from the lunch you had a few hours ago. No, it's coming from a very specialized, ready-to-go fuel source. And that fuel source is glycogen. Basically, it's this massive, branching structure made up of thousands and thousands of glucose molecules, all linked together. Its unique shape lets your body pack an unbelievable amount of fuel into a really tiny space. That's why the best way to think about it is like your body's energy savings account. It's like having cash on hand for when you need it, fast. Now, this energy bank has two main branches, and they have very different jobs. The liver, well, it acts like the body's Federal Reserve. Its purpose is totally selfless. It stores glycogen just so it can release glucose into the bloodstream, making sure your brain and all your other tissues have a steady supply of fuel. Your muscles, though, they have a personal account. The glycogen stored in there is for one customer and one customer only, the muscle cell itself, to power its own contractions. The real genius of glycogen is its shape. Picture a tree. It's got a main trunk, sure, but it also has countless branches. This branching is the secret to its speed. It creates hundreds of different endpoints where enzymes can get to work. So when you need energy right now, your body doesn't have to start at one end of a super long chain. It can just grab glucose from all those branch tips at the exact same time, delivering this massive, rapid flood of fuel. Okay, so let's move on. Let's look at how this withdrawal process, which is called glycogenolysis, actually works. How does the body get that glucose out of storage and ready to use in just a split second? It all starts with a star player, an enzyme that acts kind of like a pair of molecular scissors. It snips off one glucose unit from a branch. But here's the really clever part. As it snips, it also slaps a phosphate tag onto it. Think of this tag as a ready-for-use sticker. Then a second enzyme comes along and just slightly adjusts the position of that tag, creating a molecule called glucose 6-phosphate, which is the universal, ready-to-burn fuel for the cell. So what happens to this tagged glucose molecule? Well, this is where the liver and muscle go their separate ways. In the muscle, that phosphate tag actually traps the molecule inside. It gets immediately fed into the energy-producing pathway. But the liver has a special trick up its sleeve. It takes that tagged glucose to a specific part of the cell, the ER. Inside, there's a unique enzyme that acts like a key, unlocking and removing that phosphate tag. This creates pure, free glucose that can now be exported into the blood for your brain and other tissues to use. And this is exactly why your muscles can't share their stash. They just don't have this special key. For the muscles, this whole process is incredibly efficient. There are two huge advantages here. First, when glucose comes from your blood, the cell has to spend energy to stick a phosphate on it. But when it comes from glycogen, it's already got one. That's a direct energy saving. Second, like we just saw, that phosphate group acts like a lock, trapping the energy source right where it's needed most, inside the contracting muscle cell. So, what about those intersections, the branch points? Our main enzyme is great on the straightaways, but it stalls when it gets too close to a branch. To solve this, the cell brings in a specialist, remodeling enzyme. This enzyme does two things. First, it acts like a construction crane, lifting a short section of the branch and reattaching it to the main chain. Now, only a single glucose stump is left at the branch point. So second, it uses a different tool to snip off that final stump. And just like that, the roadblock is cleared, and the main enzyme can get right back to work on the newly straightened chain. Okay, so the crucial point here is that this powerful system of energy release can't just be running all the time. That would be incredibly wasteful. So let's explore the really sophisticated controls that manage our energy bank account, making sure we only make withdrawals when we absolutely need to. To understand this regulation, we need to talk about something called an allosteric enzyme. This is basically an enzyme that has other regulatory sites in addition to its main business end. When molecules bind to these other sites, they can change the enzyme's shape, either cranking up its activity or shutting it down. Think of it more like a dimmer switch instead of a simple on-off button. And our key enzyme, glycogen phosphorylase, is a master of this kind of fine-tuned control. Let's see how this works in the muscle. The cell's energy status sends these direct signals. If AMP levels are high, that's a clear signal that the cell is running low on energy. So AMP activates the enzyme, basically shouting, "We need more fuel." On the flip side, if ATP levels are high, it binds and shuts things down, saying, "We're good, we have plenty of energy, stand down." It's a beautifully responsive system. Now, what's really fascinating here is how this shows the different priorities of the liver versus the muscle. The muscle enzyme is an energy sensor. It only cares about the cell's internal ATP levels. The liver enzyme, though, is a glucose sensor. Its job is to manage blood sugar for the whole body. So when blood glucose is high, glucose itself binds directly to the liver phosphorylase and shuts it down. Why? Because it doesn't need to release anymore. The liver enzyme is totally insensitive to the energy state within its own cells. It's all about serving the rest of the body. Beyond these local signals, there's a master on-off switch, and that's phosphorylation. In response to hormones, a special enzyme comes and attaches a phosphate group onto our phosphorylase enzyme. This single chemical tag acts like a power button, flipping the enzyme from its default, less active state, what we call the B form, into its supercharged, fully active A form. Then, once the emergency is over, a different enzyme comes along to remove that phosphate tag, switching the enzyme back off. It's a precise and fully reversible way to control the entire system from a distance. Okay, we've spent a lot of time on withdrawals, but a bank isn't much good if you can't make deposits, right? So now that we've covered glycogenolysis, let's look at the other side of the coin, glycogenesis, the process of building our glycogen stores back up. You might think the body just starts sticking glucose molecules together, but it's a little more sophisticated than that. To be added to a glycogen chain, glucose has to be activated first. It gets attached to a carrier molecule called uridine diphosphate, or UDP. This forms UDP glucose, which is the official currency for making glycogen. It makes the glucose molecule reactive and ready to be donated to the growing chain. So the building process needs a foundation. A special protein acts as a primer, laying down the first few glucose blocks. Once that's ready, the main builder, an enzyme called glycogen synthase, takes over. It starts adding our activated glucose units one by one, building a long, straight chain. But we need branches, so a third enzyme, the branching enzyme, comes in. It cuts off a piece of that long chain and reattaches it to the side, creating a new branch. This process just repeats over and over, building up that massive tree-like glycogen molecule. This raises a pretty critical question. How does the body stop itself from building and breaking down glycogen at the exact same time? That would be like trying to fill a bucket that has a hole in it, a huge waste of energy. Well, the answer is an incredibly elegant solution summarized perfectly by this principle. And this illustrates that principle of reciprocal regulation beautifully. Remember that master switch, phosphorylation? When the body needs to release energy, a state signaled by the hormone glucagon, it triggers phosphorylation. Get this. This single event activates the breakdown enzyme, phosphorylase, while it simultaneously inactivates the synthesis enzyme, synthase. Conversely, when you've just eaten and insulin is high, the signal is dephosphorylation. This inactivates breakdown and activates synthesis. It's a perfect system that prevents the two pathways from fighting each other. So we've seen how elegant and how tightly controlled this whole system is, but what happens when a piece of this intricate machinery is broken? This brings us to a group of conditions known as glycogen storage diseases. Glycogen storage diseases, or GSDs, are genetic disorders where one of the enzymes we've been talking about is defective. This could be an enzyme involved in the breakdown, the synthesis, or the branching. The result is that glycogen either builds up in massive amounts in tissues like the liver and muscle, or it has an abnormal structure, which makes it pretty much useless. Let's look at one specific example, just to see the devastating domino effect. If that special liver enzyme, the one that removes the phosphate tag, is missing, the liver simply cannot release its stored glucose into the blood. This leads to severe low blood sugar, especially when fasting. Meanwhile, all that tagged glucose just builds up inside the liver cells. With nowhere else to go, the cell desperately tries to get rid of it by shoving it down another pathway. But that pathway gets overloaded, producing a flood of byproducts like lactate, which can make the blood dangerously acidic.
This really puts the importance of glycogen into perspective. For about the first 24 hours of a fast, your liver's glycogen stores are the main thing keeping your blood sugar stable, which is absolutely vital for your brain. After those stores run out, the body has to switch to a more costly process, making new glucose from other sources. In really prolonged starvation, this even means breaking down muscle protein for fuel, which is obviously not sustainable. Glycogen is our critical first responder fuel reserve. And so we see this incredibly dynamic system of deposits, withdrawals, and complex regulation that allows you to function, all running invisibly inside your cells. As you go about your day, this elegant energy system is just working on autopilot. It kind of leaves you wondering, right? What other amazing, intricate biological processes are happening right now just to keep you going?